Hi, Paul.
Morning.
Welcome. We're just giving you a couple minutes to let some more people come in. We tend to have a lot of last minute arrivals, and then we'll get going.
No problem. Did you ask something else? I think you froze for a second.
Oh, sorry. My Wi-Fi is a little bit spotty. I'm just going to give a brief intro as everyone's coming in, kind of an overview of everything, and then hand it off to you.
Super.
Hey, Paul, this is Jenny. I'm the director of our program. I just wanted to say thank you for taking the time to talk with our students today. I think they're really excited. So
Great. Yeah, happy to do so.
Hi, everyone. To everyone that's here, we're just going to wait a couple more minutes to let a few more people trickle in, and then we'll get started. All right, so it looks like our people coming in are trickling down. So we're gonna get started now. So first of all, hi everyone, and welcome to the Statistic Tutoring Center's final installment of our Statistic Bootcamp workshop series. Just to give you all a brief overview of what we're gonna be doing today. First, we'll hear from Dr. Paul CarDonna about making effective, visually appealing scientific figures. And then we'll take a quick break, probably somewhere around 10 minutes. And then we'll follow with Dr. Hannah Carroll leading a hands-on workshop on how to create publication quality plots. Also, please note that we're going to ask everyone to fill out a brief survey when you're done. So I'll stick that link in the chat now for you to save for later, and also share it again after each presenter for anyone that has to leave early, as well as anyone that might come in late. So before we get started with Dr. CarDonna, I'd just like to give him a brief intro. So Paul CarDonna is a population and community ecologist based at the Chicago Botanic Garden and Northwestern University. His research investigates the interplay among species interactions, population dynamics, and community patterns, mostly with plants and pollinators. He tends to ask research questions from a basic ecological perspective to better understand how plant and animal populations and communities operate in nature, but with the ultimate goal of improving conservation efforts in light of various global changes. Most of his research takes place at the Rocky Mountain Biological Laboratory in Gothic. So with that in mind, welcome, Paul.
Thanks, Hunter. And thanks for inviting me. It's always really fun to get to I think one of the great things about being a scientist is you get to do science has all these sort of demands on your brain. And one thing that I really like doing is making figures. It's a really important part of my workflow. And as a result, can you all see that okay? Okay, super. Oops. Let me. Despite the fact that I teach in Zoom, it's still always some sort of challenge. Okay, so yeah, over the years, I've picked up many visual design principles that I found quite effective. And at least early on in my scientific career, I had a couple friends that I would talk to, and we just thought we were sort of, picky about figures and things like that. And we'd sort of made up these terms to sort of describe what we're talking about, like breathing room or this and that. And then I got to take a visual design workshop with someone who spends all their time doing data visualization with scientists. And it was really sort of an excellent experience because so much of what I had been thinking about and trying to do and practicing, there was actually some principles behind that. So that's what we're going to be talking about today. Just a handful of little tips to sort of make your figures do what they're supposed to do, say something and communicate some science clearly. So let's get to it. Oh, little fun fact. So I'm in Chicago right now. We have a good amount of snow, sort of different scale to Colorado in the mountains, but my daughter, 4 years old, is sitting on the pile of that snow pile in our parking lot. It's like 12 feet, which is pretty cool. So she comes out to Gothic in the summer too, but she never gets to see the snow. And to sort of prove to you that I actually do work and I'm familiar with where you all go to school, here's me and Zoe in the field counting some flowers last summer. Okay, so the first thing is, if this slide sort of horrifies you, that's a good thing. A lot of times when I start talking about visual design, people sort of they kind of come to the table with a little bit of baggage about what they think I mean, as if we're sort of like making figures pretty, or like making, using clip art or whatever, you want to sort of like doctor up something so it looks, quote, pretty or whatever. But that's not really the point at all. And we're trying to keep things simple. So while we're really focusing on figures, you can apply these concepts to like making a presentation as well. Okay, so the first disclaimer here is that the goal of this workshop and my little segment of it is to improve all of our visual communication skills using some simple guidelines. I'm not saying I'm the best or this is the only way to do it, or that you can't, if you disagree with me, you're wrong. What we're trying to do is improve our figures so they're communicating more effectively. So that's the goal there. And I'd like to point out that a lot of this material has been shared with me. I can never say his last name, but this guy, Martin, at the University of British Columbia Genome Center, he was the person who led the workshop I went to first. And so just want to say thank you, Martin, for sharing the materials that I've now sort of modified quite a bit, but it's based on some of the things we talked about several years ago. So the first thing that I don't hear as much anymore, but I remember some older professors saying to me when I was a student, let the data speak for themselves. And that was sort of like seemed as if that was the epitome of like, a good figure or good science. Like just let the data do the talking and you shouldn't have to worry about anything else. And so in my own sort of experience, what, my interpretation of this now is like, okay, sure, let the data speak for themselves. Here you go. Here's an Excel file with a bunch of flowering phenology data or the timing of flowering across, 45 years. We got all sorts of species on the left-hand axis. There they are speaking to you. What can you see any patterns here? No, you don't have to answer. That's okay. But the point is, if we let the data do the talking, they don't really tell us that much because each data point wants to get, its own little special moment in the limelight. So we have to sort of figure out what the data are saying. So here's sort of a little baby example. We've got some numbers over here sort of representing some data, some values that we've measured. Now we could change those to maybe circles or something, something to communicate the value. The number themselves is sort of an abstract, we have to sort of translate the number 13 into a value of some quantity. So what we've done here, just as an exercise, is made them into circles of different sizes. And now we're already starting to see some patterns in the data that we couldn't see before, that the data weren't really saying on their own. We could sort of arrange the numbers in a different kind of way, grouping them in bins to perhaps reveal something else. And we may do something a little bit more traditional, using these bins here, whatever the bins are, adding a little dotted line to maybe emphasize something that we want. One thing that nothing jumps out when you just look at the numbers, the little dots in the second version, you can see that there's basically 4 big dots and a bunch of small dots. But then when we start to organize them in certain bins, we see that there's really, there's some small values, some large values, but there's this where we have we have only one value for this sort of medium bin, which might be really important to communicate, none of which would be communicated with just the numbers themselves. So each of these examples is illustrating something different, albeit nuanced, but something different about the data. And as scientists, we really want to think about what it is we're doing in a figure and what we're actually communicating. And so I like to think of this general process, or sort of thing number one, the concept of reduce to reveal. So we're sort of reducing the complexity in that Excel file or a CSV file or text file, and we're reducing that complexity to reveal a pattern in the data. When we're doing statistics and things like that, we're trying to sort of not just look at the figure and sort of extract something rigorous about a pattern, but we do want the figure to reflect those statistical analyses. And this is a way to sort of do that. We're reducing the complexity of the data to reveal the pattern we're trying to show. So we'll sort of go through some simple examples. Some of these I don't want you to worry in the slightest about what the values mean. Just sort of glaze your eyes to that and focus on sort of the visual. And one sort of thing I like to do is what sort of jumps out to me initially about a figure before I sort of digest any of the actual science. And so we can call this sort of set of plots a bit confined. We've got these 6 panels. We've got some values, we've got some number, we've got some little titles for each panel. We've got all these open dots. And if you're just kind of looking at it, to me, what happens is I kind of ride the wave of the points here. And I'm like looking across the panels as if I'm supposed to be getting something by like connecting all the dots, like I might do with my daughter. We're doing a little art time. So it's a bit confined. Now, again, don't worry about any of the values here, but we sort of have this baseline value indicated by by 2 on the y-axis. But what we can do with the hollow circles, we're sort of emphasizing the trend, those lines, and with the dot, with the filled in circles, we're emphasizing the data. And so here's sort of a simple case where we might make a decision about filled circles or open circles, and that really ends up emphasizing either the trend or the data points. And either of those could make sense depending on what your goal is or what you're trying to communicate. So we're sort of cleaning up the figure here just with some pretty minor things. We'll get into this with some other examples, but just to start to set the stage here. So another thing that I come across when talking about data visualization, although I feel like this is also starting to change in terms of people's perception, is that, we've got science, and we've got this really fine line, and then lies and manipulation. lies and manipulation being we're deleting data points and we're moving them around and we're making, in the process of making everything pretty, we're sort of lying and manipulating our audience so they see exactly what we want to see. And we're really just trying to deceive them and show them what we want or something like that. Whereas on the other side of the spectrum, we have science. And of course, I'm being dramatic here. It's so pure and there's no altering or tinkering, it just, the data speak for themselves and we're good to go. And so there's almost sort of this perception, which again, I think is changing a bit, that if we use the sleek font on the left, we can get away with 1 + 1 = 3. Because people are like, oh, look, it's so sleek and put together. But if we use a sort of like chalkboard style thing, that's sort of, that's the truth, right? But I think that's really unfortunate because I would say there's actually, a giant wall of intellectual integrity between science and lies and manipulation. You got to climb a pretty big wall, the big wall of intellectual integrity, before you jump over into lies and manipulation. So as scientists, we're trained, we don't want to be lying and manipulating our data and things like that in a way to confuse someone or make them think there's a bigger pattern than not. And intellectual integrity is what sort of helps us, of course, stay away from the sort of, danger zone there. So when we're doing data viz, we're far from this area of lies and manipulation is the main point here. And when you're crossing that line, because you still have to start doing some pretty funky stuff. So if you were worried about that, probably not, because you're taking a workshop on this stuff, then here you go. All right, so my little toolbar, let me just hide the video panel. Oops, wrong one. There we go. Now I can see. Okay, so here's some genomic data. It's not my data, but this was shared in some slides. We could say it's information rich, but uninformative. So we'll go through a few examples here. And I think this is a really good way to think about your figures. In this case, it's a lot of information. We've got all these chromosomes, sort of, chromosome numbers indicated by different colors, and it looks interesting, but there's absolutely no information that's being communicated here. I have no idea what we're supposed to get from this figure. Like, okay, there's variation in chromosome numbers. So plenty of info, but a total dud in terms of being informative. Here's a case where we have, it's informative, but not that information rich. And this is fine. A lot of times we have a need to show something pretty simple like this. But in the end, there's not a ton of information, especially if we go between the two. We've basically got the mean and error of two sort of, treatment conditions here. That's it. There's not a lot of info being shown on this figure. Here's for my own research, doing ecological networks. This is a common thing that people like to make, but I would argue that despite the fact that it's information rich, it's maybe somewhat informative. In this case here, a paper by a colleague of mine, Laura Burkle, who actually did her PhD at the Rocky Mountain Biolab, so just up the road from y'all. In this case, we can see, okay, there's some things popping out. There's these red things, and there's these red lines and red boxes and blue. But for the most part, it just kind of looks interesting, but we're not getting a huge amount of info from it. Here's the case from a science paper. I would say it's information rich. It's a lot of studies that have gone into this, but I would argue it's maybe a bit confusing. You look at this, and the first thing I think of is like spaghetti and sauce. And then the second thing I'm thinking of is, why do we have histograms on the top and sides? And then, oh, there's a black line in there too, and a dotted line. And then we've got a sort of bar plot inside. It's like, there's so much information here. I don't even really know what I'm supposed to look at. And I don't think I have the point is we're looking for a hump shaped pattern. And so that's what this black line in both of those plots, that's really the main thing they're trying to illustrate. And I think with all this other stuff, they've made their figure quite a bit more confusing, when they could have reduced some of the complexity to end up showcasing the pattern that they really want us to understand. Doesn't mean we need to get rid of everything here, but we can sort of play with emphasis to communicate something a little bit better. Here's a paper that I got to contribute to. Again, don't worry too much about the what it actually means, but I would say there's a lot of information here, and I would say it's relatively informative. You look at the figure, and without even knowing what you're looking at, you can see sort of differences between the red and the blue. And to me, that's the sort of the first checkbox I want is for someone to look at this and kind of be able to, if we stripped away all the axes, so all you saw was the actual figure itself, you could at least make up a story like, oh, for the top panel, the red is greater than the blue. And for the bottom panel, for example, the red is lower than the blue. So a lot of information communicated here that's relatively easy to digest. And then the sort of data points, which we don't want to emphasize, are sort of fall to the back at least a little bit, being gray open circles. We really want to emphasize the trend here. Here's sort of a different style figure. This really isn't showing data per se. It's sort of a conceptual diagram. So it's I would say maybe information rich. It actually is digesting visually four different figures sort of put into one with multiple panels. And I'd say it's relatively informative, but this needs to go with the actual data, or else on its own, it's a little bit too simple. But it helps to communicate some figures that are otherwise fairly complicated to digest. So in this case, we're sort of doing a little bit more art and design to help leverage a figure or the sort of digestibility of a more hardcore figure. Here's some data from a colleague. And there's a lot of information here. And we won't go into the details, but I think when you look at it, you can see with this cloud of points, tons of values there from these simulation models. We see that the black dot represents the sort of overall mean. It's not in the coexistence region. And then we see this big cloud of points, which sort of helps to communicate that most of the simulations of these two species competing against each other are falling in this sort of upper area where one species wins. And we have basically none of the dots from any simulation falling into the coexistence region. So here's sort of just one more figure, one from my own work. This is sort of a conceptual diagram, so not something you'd have to sort of make this, but sort of describing a series of simulation models in a visual way. And what I found was my co-authors and other people that were reading the paper were struggling to understand what I meant by all of the sort of hypotheses in our simulation models or what they were doing. So I made this visual, and the sort of what ended up happening was people stopped asking questions about how the simulation models worked. So of course, I don't expect you to digest all of this, but by making a visual to then be paired with the text, which is on the right, basically a couple pages of text in one figure ended up sort of clarifying a series of points I wanted to make. So the design of the figure took quite some time, but it ended up, I think, saving a lot of people from getting confused over a really important part of the paper. So those are just some examples of sort of thinking about this having an information-rich figure and whether it's informative or not. And I think that's a good place to be. Not everything needs to be information-rich to be informative. In fact, we can have a lot of cases where we have something that's information-rich that's uninformative. So you want to kind of find the sweet spot depending on your message. Which brings us to the next part, which is really thinking about the message that your figure is communicating. So you really don't want your figure to look like a Where's Waldo book. And I would argue that, unfortunately, there's plenty of figures in the scientific literature that are kind of like a Where's Waldo book. I would rather, I love figures, but in many cases, I'd rather just read the text because I have no idea what's going on in the figure. You know, you have to stare at it for a long time, go back to the text, back to the figure to really make sense of it. To me, that's a figure that's not doing its job. So your figure should not look like this. Another book series you might be familiar with, maybe you can tell that I have a child, is I Spy. You know, your figure should not look like an I Spy book. Don't make your audience try hard to figure out what the message is in the figure. You need to communicate it through effective visual design. So one of the points here is deliver your message with a necessary degree of supporting detail with as little ink as possible. So keep it simple. We don't need to have a bunch of clutter all over the figure. So by keeping it simple, we can really sort of deliver a message more effectively. And I always ask myself, if a figure has a message, what message is the figure sharing? And that's why I say, when I like to look at a figure, we actually did this in my lab group last week. Someone showed a figure, and I had to confess when we were talking about it, I actually forgot to read the caption. So my comments were based on looking at the figure without the caption. And for the most part, I could understand what the figure was saying without reading the details. And I think if you can do that, that's really great, because your message is coming through without anything else except the figure. And so my take on papers and things like that, or a conservation report, whatever it is that when someone's reading your paper, the figures alone should illustrate a fairly complete story. So when I'm writing up my science, I want there to be a series of figures that, whether they're showing a pattern or a lack of pattern, that if you read the title, read the abstract, and looked at the figures, you'd be like, okay, I think I buy it. And so that's sort of the high bar I want to get to, is that the figures can really tell a lot of the story on their own. Now, of course, I'm not saying that only the figures can do that, but if you can get a pretty good complete story from the figures on their own, I think you're doing a good job illustrating your results. So you don't have to shout anything out, but why don't you try and count the dots really quickly? And so maybe you have some number, but the point is, if we rearranged them, you'd be able to count the dots way easier. You can sort of look at this and you know there's 16 dots almost instantly, because it's arranged in a really intuitive manner. If we wanted to emphasize that there were, 4 groups of dots, still 16 dots, but just by moving them apart, you're like, oh, 4 groups of 4, 16 dots. So by playing with just the spatial arrangement here, of course, this is what you wouldn't do this with an actual figure, but these are concepts you can think about. By giving some space here, we're making the viewer is sort of looking at 4 groups of 4 dots. Maybe the spatial arrangement is really important, and we want to emphasize that most of the values or something like that are in the center. Maybe these points on the edge are outliers that we know are, we had some whatever. Our grandpa came out and helped us collect some data, and we wanted to point out that we don't really trust those. Or maybe we want to point out that there's really just a few dots away from the center. So perhaps the outliers or something like that, or these extreme values relative to the average, are actually really important, and we want to emphasize them for some reason. Maybe one dot is very special. Now, again, this is sort of, we don't have a lot of info except 16 dots here, but perhaps this is, here's your field team and someone has COVID, stay away from that person. Or perhaps everyone's had COVID and you're the only person left who hasn't. And we want to emphasize that our field team has done a bad job wearing masks and things like that. Who knows? But as you hopefully what you can see is, just by little sort of moving things around in space, maybe those are panels of a multi-panel figure. Maybe these are your actual data points and you want to code in some color to illustrate something. You can get a lot just by a little bit of change. So when it comes to figures, and as I've sort of touched on with the last set of slides, each piece of a figure communicates something. So in the past, and in the last slides, we're sort of talking about just the spatial arrangement or the colors or things like that. Each of those things is communicating something different. But if we think of figures that are a little bit closer to home in terms of the types of things we plot as scientists, Here's sort of a simple bar plot. Really all we're seeing here is quantities. You fill up the bar and one is bigger than the other. So not too much going on there. But if we add the little error bars, it's not that necessarily error bars are required by some written law in science, but they do tell us a lot of information. And by adding them, we're now sort of providing some info on the spread of data. Now, in general, I would advise not using bars with error bars like this because it's kind of a funky way to do it, unless you have proportions maybe. So maybe this is a little bit better. We have a mean value with some error about that mean, maybe the average size of a bumblebee in a warm year versus a cold year. Or maybe we want to use something like a box plot where we have sort of central tendency shown by that thick line in the middle. We have most of the spread of the data indicated by the box, and then we have sort of most of the range of the data when we include those the tails on the box plot. And so one of the points here is that each of these this is all the same data, but these are sort of emphasizing different parts of the data and communicating different things about the data. You can almost imagine the distribution of the data with the box plot, whereas we don't have quite as much info when it comes to just the dot and the error bars. And we sort of have a funky version of it with the bar and the error bars. So all of those things, depending on what you want to do, can be really important decisions for communicating your message. Now, this is sort of let's just use another example with lines. I've seen papers like this or figures like this in peer-reviewed publications. It's sort of like pickup sticks. Okay, cool. Maybe each color represents a different species and we're looking at some trend, but all you really see here is that there's a bunch of stuff going on, which usually means you probably can peel back a layer of the visual design onion to help illustrate something more effectively. So maybe we want to point out that one of these, maybe this is the abundance of an invasive species in a community. And that species abundance is going up over time, whereas all the native species are going down or mostly down or something like that. But still the same data, same information. And just by changing the color of the lines, we can really draw attention to something that is maybe quite important. Maybe we want to point out, maybe we're just trying to say, these are the trends that are meaningful statistically or whatever. And the gray ones that we want to kind of fall to the back, those are the ones that there's really nothing going on. So by changing the color there, we're emphasizing the sort of strong patterns and de-emphasizing the ones that really nothing's going on. Or maybe we want to actually not have all of our noodles on the same plot and spread them out in separate panels. We could have this all gridded up so that they're all in their own little squares, but this sort of shows these are all the same values here, that we have sort of some steep values, we have some moderate values, we have these sort of maybe statistically non-significant values in the sort of lower panels there. And then we have this really sort of striking positive value or something that might be really important for us to know. So all of those same data, very different sort of story that we might tell. And then we won't spend as much time here, but here's some dots. Again, the dots, the black dots themselves emphasize the data points. That might be really important. By making them gray and putting a trend line, we're emphasizing the trend. By highlighting that sort of low point there in red, maybe that's an important outlier. Or maybe these are two populations or two sexes or something like that. And we see by color coding that additional factor, sex or whatever, we see that the pink dots are all high and the blue dots are all low. And maybe we really shouldn't be showing the data as a trend line because most of it has to do with the pink dots being up high and occupying that space and the blue dots being down lower. So all of these sort of visual design decisions can really change the meaning of your figure, and you should be thinking about these. So I think something that's important to keep in your thinking cap is that it's pretty easy to make something complex, to make complex things appear complex. right? You know, a network diagram that I showed you, whoa, look, complexity in nature, great. But if the message of your figure is that it's complex, most of the time that's sort of a cold meal. I think we can do better. So here's a pollination paper that I've read a few times, and I still have never quite managed to figure out what the sort of figure means, panel B. It's a complex paper. We've got a sort of three-dimensional plot here, and I've never quite been able to figure out what those sort of shapes mean or exactly how I'm supposed to extract it. It's just too confusing. So complex things appear in complex. It's a lot more difficult to explain complex things simply, but that's what we're trying to do with a figure. If you plot any figure, you're taking a complex data set and boiling it down to something a little bit more simple. But that might not be that informative in the end. So what we're really trying to do is break down complexity to make things simple. And so I think if you're spending time before you get to the R dashboard or whatever your favorite program is, you know, think hard about what your figure is really communicating. I often draw out my figures in a notebook before I start plotting them because I want to know what I'm trying to communicate, you know, at least conceptually before I jump into dealing with the data as sort of a guideline. And if I know what I want, it makes it a lot easier to figure out how to do that. Okay, so another concept here that's useful to think about is focus and clarity in your figures. So what are you focusing on, and is it clear? So here's sort of a sign that's, you know, clearly pointing out to you that it has sharp edges, but turns out that the bridge is out ahead, which is probably a lot more important and, you know, is not quite clear from the way we've sort of set up the sign here. Here's just a photograph to sort of illustrate this point. A pretty cool photograph. We've got these swans. They're very bright. You know, black and white photo. This person with a black jacket feeding them. And in terms of focus and clarity, as a sort of visual design principle in general, if we make this just a little bit gray, it really changes sort of that how strongly you react to that sort of contrast there. And so these are things that we might want to de-emphasize something like this, or we might really want to make it quite striking like that. So you want to be thinking about that stuff when it comes to dealing with your data, so you don't lose points or that you don't sort of end up distracting someone with something you don't need to. So here's another way of thinking of this. You know, we've got this important thing that we want everyone to look at, but, you know, it's much more interesting to look at this look here text on the right. And, you know, by making them gray, it sort of changes it a little bit, but maybe if we sort of did something like this, now you know what to look at. That red point really draws your attention pretty quickly. So here's it's fun to pick on science because sort of like the top of the top for scientific papers. Here's you know, you can see the patterns here if you look at it, but like when I look at this, okay, it's a visually interesting photograph is what I think of first. And I'm just sort of like spacing out on like, wow, that's so gnarly. Like, look at this, you know, the effects of deforestation. This is so crazy. And you're like, oh, right, there's a figure here. And it's like this Excel default, you know, you change the points to diamonds. And I don't know, it's just sort of a missed opportunity here by having the figure. We're sort of drawing a lot of attention to a figure and de-emphasizing the data, which gets lost. Maybe separating them and putting them side by side would be a better choice. Okay. You'll all be making figures in a bit, so pardon me if I've made everyone's lives harder, but don't trust the default settings. I would say this is a really good sort of rule of thumb when it comes to making figures. I'm going to assume that most of you are doing stuff in R or maybe Excel, but don't trust the default settings or what the program makes. So just because the program or the package can plot it doesn't mean it's a good idea. And it doesn't mean it's good design. So here are just some examples from R with some defaults. We've got sort of our, our noodles, our pickup sticks on the left with a ggplot example. And, we've got this gray background. We've got sort of no box around the figure. Maybe that's okay. There's this legend on the right-hand side that's, doesn't quite fit perfectly. Then we've got these in base plot, these box plots, and they're kind of like these chubby box plots. I think something that always annoys me about the defaults and box plots is their width, or in this case, their height. But the data is the ozone. And so it's interesting to me that the sort of chubbiness of the plot has more space dedicated to it than the actual box plot itself that's showcasing the data. So it's kind of a weird effect. I think we can shrink down the sort of chubbiness of the box plot to sort of emphasize the actual data range rather than this default width. And then over here with you can sort of add in these different colors, but it's kind of like I don't know, I feel like that's something my daughter would draw. We've got these like dashed lines. I think this is in lattice. But it's just kind of confusing. The feet on the error bars are gigantic. It's just kind of a messy figure. We could simplify things quite a bit. Oh, there we go. Okay. Here's a paper, this illustrating the sort of box plot situation from a colleague of mine that I don't know that well, so I don't have to sort of, defend him. But these are box plots. Look at how wide they are. Like, it looks like you sort of want to, like, if you looked sideways, you're like, oh, is that how I'm supposed to look at it? But no, you're supposed to look at it this way. And the width is so distracting to me. Like, it's so much wider than the range of data points. We could probably do some things to kind of change to make this a little bit clearer and not so distracting. So here's just some of my some data of my own. On the top is sort of what I've done to the box plots to sort of emphasize the things I want to emphasize. And on the bottom, using the same or similar colors, is what you'd get from R by default. So by cleaning it up, obviously there's a little bit more data on the top. By cleaning it up, we sort of help to emphasize the actual data rather than these sort of default bins or widths. So this is sort of something that you see a lot of examples online if you're like trying to figure out how to plot something in ggplot, and you get this sort of like gray sort of title at the top with the year, and you get this gray background, which we don't really need for the most part. Like, do we really need to be able to find exactly what the value is for each of these points? Probably not. Then we've got this blue line with the gray error bar, which is cool, but there's a bunch of stuff going on here that we could really sort of simplify. The points are black, things like that. So this is kind of a default setting. And I find that people sometimes argue like, oh, well, the default's pretty good, right? That's why it's a default. But no, the default's just there so that it's some sort of starting point. So obviously these are different charts, but here's my own sort of version of a squiggly line. I've removed the actual data points because I wanted to emphasize just the trends there. And it just is like way too much going on when you have all those points. So I've stripped away the gray background. I've sort of cleaned up the axes, and I've really tried to emphasize these different lines so that you can see that blue is lower than gray and red is greater than gray. It's a warming experiment. The other thing I think is important to know is that, R and Excel or whatever you're making your figures in, they don't know, ggplot or base plot doesn't know what you're trying to show. So you need to go in knowing what you want. And if you know what you want, you can it's a lot easier to figure it out rather than starting with a default. I mean, you often start with a default anyway, but then trying to figure out like what it is you want to show. Because as soon as you start plotting stuff, you're starting to, what you're looking at is starting to influence your own thinking process. So it's good to show up to the R dashboard with some ideas for what you want to show and emphasize. So this is for 1 year of data, some plant pollinator stuff that I made in Excel years ago. Now I don't do anything like this in Excel because it's kind of a nightmare. And then here's many iterations later what it ended up looking like as a publication, where I'll show you some iterations in a moment, but quite a different story in the end. It's hard to sort of see a lot of things here, and they're much more clear when we plot it like this. So we'll get to that in a moment, the sort of iterative process. Okay, this is, I think, a really fun one, nice and simple, and really easy to manipulate in R and base plot, ggplot, things like that. So use intuitive symbols. And so if we look here, let's imagine we've got some sort of gradient from weak to strong. What would you do? What would you put if you had an intermediate symbol? So to keep things simple here, I won't sort of call on anyone, but just think about it for a second. What might you put for the intermediate value for both sort of sets of shapes? So pretty simple. When we're down below, we've got circles, black and open. Gray would pretty clearly indicate something in the middle. Not as strong as the dark circle and a little bit stronger than the weak circle. But what do we do when we get to intermediate on the top? Like if we're in between a triangle and a circle, like a square technically isn't really in between. If you think about, the number of sides on a square versus a triangle versus a circle. So then we'd have like a polygon if we wanted it to be like legitimately in between, which would look like a circle, which sort of defeats the purpose. So the point here is don't use unintuitive symbols like triangles and circles for weak and strong. Use color or something like that, or just a grayscale that much more effectively communicates the strength, or something like that. Maybe where I'll show you an example in a moment, but perhaps the open circle is the control in an experiment, and the black circle is the experimental manipulation, or like the extreme one, or something like that. You can sort of play with other concepts here too. So here's a figure from a grad student that I spent some time working with in Denmark. And this was his cleaned up, the first, after we sort of talked about stuff, this was his first plot. And if you look at it like, okay, whatever, could be worse. But we've got different shapes for the different sites. We've got different colors for the different sites. We've got different line sort of patterns for the different sites. And I don't even know if I already said shape for the different sites, but there's all this stuff going on that in the end, I think does a lot more. Like, is it meaningful that there's a dotted line for site 3? And like, you start to wonder, if you're thinking about, are there other things we should be paying attention to here that I'm missing from the text? Whereas this is a different paper, but here's sort of a more simplified version of very similar types of data, where we've got the control and we've got 2 treatments. And, it just sort of, the lines are the same, the error bars are the same. All we're really doing to indicate differences in treatments or plot type is the color. And so this isn't quite as sort of simple as like weak and strong, but I think it does make sense that your control is the open circle, and the 2 filled in circles are different treatments. Those are sort of like the more extreme or fancy versions, like control being white. A white open circle is kind of a nice intuitive starting point. So this is some cell types. This is an example that Martin shared. And he was presented, he's at this genome center, and some of his colleagues came saying like, here's our cell types, we're trying to make this diagram or whatever, and here's our cell types. And his first response was, I don't really know, like, I don't know what these things mean. I know that cancer's bad, but there's like not really an intuitive way to know which one's the worst. He's like, so if I had to pick one, if I was supposed to stay away from one, I guess I'd choose cancer, the red one. But it's hard to say, like, which one's worse. And so this is a pretty challenging one. And I think it's a little bit beyond what we tend to do when we're making figures in R, but I think it's still a good exercise to be thinking of this stuff. So what types of symbols could we use to make this more intuitive? Now, we don't have to worry about that now, but just take a moment to imagine, and then I'll show you what Martin came up with. So his goal here was to sort of, the ones that are good or not bad should not look as intense as the bad ones. And so here's sort of his mock-up. Cancer is kind of a nice reference point. That's bad. It's black. And then immune, I think, is pretty intuitive that that's a good thing. We want immunity. So the immune cell is sort of got this thick wall on it. It's stronger, more protected than normal. And normal is sort of, nice and simple. Just a plain, it's like the default, a circle with no color. Necrotic means it's starting to die. And so here we have sort of like, doesn't quite look as good as normal, but it's not quite as bad as cancer. And then neoplastic, it's got a cancer cell in it, I think. Again, this isn't my field, but we've got that little cancer cell in there. Cancer's black in this case, so it's sort of like this strong or intense thing. And then the cancer stem cell is the worst, and that's the one that you probably wouldn't touch if you had to choose here. That one's like the gnarly one that's spraying and you should stay away from. So, okay, pretty out there example perhaps, but this is a pretty good exercise in trying to think of what an intuitive symbol might be in a complicated case. Okay, so we're coming up towards the end here, but a few more important things. Spacing and layout is a really nice tool to have in your back pocket. Align and space ruthlessly, or else. So when you're organizing figures and things like that, you want them all aligned. Sort of reduces visual clutter and keeps people on track. When it comes to sort of margins, like your plot margins, or like where you're putting a figure on a slide, or how you're arranging multiple figures, when you put things towards the edge, it often has some tension. But if it's nice and centered in the middle, we've got some calmness to it. And although this isn't going to make or break your figure, when you start to add up all these things on top of each other, if you have lots of tense sort of layout in a paper or among a bunch of figures, it starts to just kind of get in the way of a more calm and sort of digestible experience. So if you're making like a poster or designing a sort of more complicated figure, by having things like all over the place on the left, it's a bit cluttered, but we could clean that up pretty easily just by sort of laying things out a little bit more simply and sort of organizing how the viewer looks at it, rather than sort of making them go through this like a blackberry bramble or something. This is one that I like quite a bit, especially for multi-panel figures, which is pretty common. Space makes groups, and no space makes no groups. So by how you group things, whether it's bars within a figure or panels in a multi-panel figure, the spacing is a big deal. So here, just looking at these circles and triangles, we've got no space. I don't really know what you're supposed to get from this, but it's whatever, there's no space here. So you're like, okay, circles and triangles. By adding the lines, we're sort of maybe trying to direct people to look at the columns rather than the rows, We can space them out a bit, which helps to sort of emphasize the columns. We can space them out even more, and that really helps to emphasize that we want to be looking down at the columns, not across. So when you're laying out figures, keep this stuff in mind. It's important. It goes a long way, even though it's a little change. Okay, I think this might be what I have last, but figure making is an iterative process. So I can't stress this enough. Don't expect that the first, you make a default figure and you're good to go. But beyond that, it takes time and iteration to really settle on what's most important in your figure. And so here's, this is actually a few steps in from a paper I was involved with a long time ago. And then here's what we ended up publishing, which took some time to get there. I don't even know if this is what I would make now. So in terms of this being an iterative process, if you asked me to make this figure again, I'm not sure I would settle on the same thing, but I tried really hard. Here's another one that I do think I'd make the same figure, not this one, but this is sort of, well, this was draft one. And I was so stoked that I was able to do this in R years ago. I was like, look at this. I got these little dots and lines. And then I looked at it more and I was like, this is pretty messy. I don't know if I actually like this. So then I separated things out. And what I didn't like about this was that, we have these different colors and they aren't really meant to, I feel like it seems as if there's meaning in the colors of the dots and the lines when there's not really, they're just different metrics. And I also didn't like that when you look at it, it's almost as if you want to look at this trend that continues over from left to right, which is not what I want people to do when they look at this. So then I tried doing it like this and I was like, ooh, I think I like this better. But it was driving me nuts, this space between the two columns of data. And you sort of look down for 2013 and down for 2014. And I wasn't sure if that's what I wanted. The thickness of the box is the same thickness of the trend line. And so for a visual effect, that's kind of weird. Like the box of the plot is the same. It has the same visual weight as the trend line, which doesn't seem very good. But I felt like I was onto something. So then I got rid of that space. So I was like, okay, maybe that'll help with how people view it. But still, I wasn't quite pleased. Something was bothering me about this. And so after some blood, sweat, and tears, I settled on this, and I was quite pleased with what this communicated. It's sort of, I want people to look at the whole figure. I want the trend lines to be popping out and the data points to be de-emphasized, but I still wanted them there. And here you can sort of quite easily look across to see if the different years are similar, or you can look down to see how the different metrics differ. But I really wanted people to see whether the years were similar or not for the different metrics. And so they're set up in a way that you kind of look across visually before you look down. Here's another one, trying to show some variation in the range of data points. And I forget what I even did, but these are some network metrics. And this just wasn't quite working for me. And here's a closer look. The gray points are the ones that overlap with 0. The black ones don't, but this was driving me nuts in terms of showing change. And so I really wanted to emphasize, I realized, it took me a while, that I wanted to emphasize the range of data points. I wanted to de-emphasize the mean. And I really wanted people to notice that for the range, it indicates the range of values we get in these network metrics, like species richness or interaction specialization. The little white dot, which I still wanted there, is the mean. And then the gray dot is what we would get if you didn't sort of break down the network into small pieces. And so the point is, the gray dots in most cases don't even fall within the range of values you get for the weekly values. So by looking at it, you sort of, one of the points is this range of value of points. There's a lot going on. There's a lot of data when we consider the full range that's not really captured by just the mean. And the mean of say what happens from week to week is not the same as what happens over the entire summer when it comes to network metrics. So this was all done in R, and it took me some time to figure out what I was trying to say. And this was the message I wanted, but it took me a while to figure out how to show this visually. Oh, okay. We got a couple of minutes. We can blaze through this. Don't be misleading. So here's one example of potentially being misleading. The relative size using disk area, and relative size using disk radius. Otherwise, we've got the same. If the numbers are the values we want to show, we should really be thinking hard about what they're doing, because using disk radius makes 10 look a lot smaller than 30 instead of using disk area. So often using circles to show sort of an amount is a little bit problematic. You really want to make sure you're not being deceptive when you do this. I'm not saying it's wrong, but just think hard about it. Partial range versus the full range of data. This is an interesting one to discuss and to think about, because sometimes we could be quite misleading if we're chopping off most of the range with the indicated by the red bars there. But at the same time, sometimes we could be quite deceptive by showing, quote, the full range. If showing everything is de-emphasizing some patterns that are actually really important, because our measurement precision is so fine that these differences are quite meaningful. So typically you don't want to be hacking off your axis range to just show the differences. You really want to think about what's the range that's showing you what you need to be illustrating. So usually a good rule of thumb is you want to show the data range at the least, and you don't want to be cutting things off that would include data that you measured. So I have some other examples here, which I can share the slides, but I'll just go through them real quick. So here are some figures for you to think of. I'm not saying whether they're good or bad, but you could work through these and come up with a visual design solution to try and make them better and communicate more science more effectively. So those are all the ones there. Like this one here, we got way too much going on. I don't know what we're supposed to see. But with that, I'll leave it on the Where's Waldo slide. And I want to go into, oh man, we're already 1 minute into the next thing. Sorry. But take a break. Hopefully this has been useful. I definitely recommend, before you start making figures, spend just a couple minutes at the least thinking about what you actually want to show before you start getting hot and heavy in R or whatever you're using. Okay, I'll stop.
Thanks, Paul. We're going to take probably just a 5-minute break. I'm going to stick that survey link in the chat really quick for anybody that's going to hop out. And thank you all for your time so far. So we'll be back in 5 minutes.
Did you want me to stick around at all, or you're moving on? You got a tight schedule, so I don't want to. I don't know if anyone has questions, or they can email them to me as well.
Yeah, if anybody does, they can absolutely jump in now. Yeah, and you're happy, if you want to stick around for the next one, absolutely do. And if not, the other step to go to, we understand. And thank you for your time.
Cool. Yeah, I'll have to peel out, but I can stick around if anyone's got any questions right now. Which is always tricky when you're in Zoom and everyone's got their camera off and are muted, but so it goes in the Zoom days.
Paul, I actually have a question for you around kind of the iterative process bit of everything. How do you work with, especially when you're doing collaborative work and people have different ideas about, what design ideas are better? Like, how do you work around that, and how many versions of things do you end up making when you have larger groups to work with? Yeah,
great question. So I've been in this situation a few times now, and I sort of will let people do I don't like to sort of jump in and be like, hey, I know the most about figure making or whatever, I spend the most time on it or something like that. But my goal is that by making a good looking figure, you're communicating the science everyone wants communicated better. So I kind of let things see what happens, what settles. Like if someone's doing most of the analysis and is going to make some figures, I wait till they make a version, and then I'm like, oh, okay, I got some feedback on the figures that I think could make them better. Or I just do them myself, but I make it clear why, what I'm doing and why, and I might even show a couple options. And I think this comes up a lot. Like, what do you do with people who have different opinions about visual design? And I think there's a little bit of a misconception that, you know, it all comes down to opinion. I think when you get to the end, there can be sort of color choices, things like that are certainly an opinion. But when it comes to the core of communicating the science, and sort of trying to reflect the statistical analysis, there are ways that work better than others. And if someone proposes something that's, you know, I don't like to be too tied to my solution. So if someone's like, oh, have you tried this? And they present it or draw it out or whatever, and it's good, then it should be clear why it's better. And then let's go with that. So that's kind of my approach. I try and sort of, you know, when someone shares a figure, if it's really doing its job, it should be pretty clear. And if it's not, then it's nice to sort of talk about like, oh, why did, you know, maybe we should change the colors here or whatever. I was noticing this when I think we really want to be emphasizing the trend. And I think that's a good way. If you fall back onto the concepts and what we're trying to communicate, it's much easier to convince people of why we need to change a figure. So yeah, in my own collaborative work, I mean, sometimes you got to draw the line, but I definitely, whether people like it or not, I make lots of comments on the figures, or I just ask to make them myself. And so now in some collaborations, like that's one of my jobs. Like, okay, Paul will make the figures eventually, which is fine. But I like to sort of make it a collaborative process and let people have input and comment on what they think is popping out.
I have a kind of similar question just in terms of like drafting and revising figures. Like I think a lot about like GIS classes I've taken, and they're like, you should be able to show a map to somebody and like it should speak for itself. But I'm wondering, like with figures in scientific publications, like you're often creating these figures for a specific audience. And so you'd want it to be able to like speak to the audience, but not necessarily like a general audience. Is that like a correct approach to creating figures, or do you think that's like limiting the scope of your science communication?
I think it depends again on what, you know, some results and some data are going to be inherently more complex and maybe are tailored to a particular scientific audience. But my approach is always, if a kindergartner could look and be like, the blue boxes are bigger than the red boxes or something like that, then great. If it's accessible to a kindergartner, then it's certainly going to be accessible to a scientific audience. So I think you want to be careful with where you're, you know, when is it okay to have additional complexity? And I think it can be a bad excuse that it's, you know, scientists are trained to look at figures or something like that. You still want to find that sweet spot where you're getting in, you're making it as information rich as it needs to be to communicate what it needs to communicate. And so I like to think of the optimum amount. I'm not saying I know what it is, but we can imagine there's some theoretical optimal amount of complexity in a figure that you hit it and you're like, oh, it's got all the info for everyone, the kindergartners or whatever, the high schoolers, all the way through the experts. And I think for GIS stuff, you know, the point that you're making, like someone should look at the map and know what it means, it's a little bit more complicated with a data figure, but it should be similar, that you look at the figure and you're like, oh, there's a positive trend. I don't know what the positive trend is, but I see there's a positive trend. Something like that.
Thank you.
Yeah, great question.
All right, thank you again, Paul. We're gonna move into our next presentation now and more of the interactive portion of this. So I'd just like to briefly introduce Dr. Hannah Carroll before we get moving on her part. So Dr. Hannah Carroll is an IRA CDA at UCLA postdoctoral fellow and an early career fellow in UCLA's Center for Diverse Leadership in Science. She received her PhD from Iowa State University with co-majors in ecology and evolutionary biology and environmental science. Her dissertation research focused on the application of big data techniques to late quaternary paleoecology. Her postdoctoral work centers on the application of cutting-edge big data techniques and novel statistical approaches to the calibration of paleoclimate signals and the exploration of plant functional ecology as a new paleoclimate tool. So with that, the floor is all yours, Dr. Carroll.
Thank you so much for that introduction, and thank you for inviting me to be part of this workshop. I really enjoyed Dr. CarRonna's presentation, and we're going to actually build on a lot of the stuff he was talking about. To get us started, I have shared an RStudio Cloud link in the chat. I'm also going to put a link. I'm also going to upload an R script and a CSV for us to work on. Anyone who's working on RStudio Cloud can hopefully click that link. It will ask you to create a free account so that you can access the resources I've put up there. And so I have a paid account so that everyone can access what I'm writing. I'm going to start with a brief presentation, and that will hopefully give you time to open up that link and get an account ready while I talk to you. Before I do that, I'm going to go ahead and put the local files in the chat. Give me just a second to pull those into the chat. I didn't want to do it too soon and have them lost. Okay, so I'm going to upload one R script and one CSV. Most of the data we're going to be working on is going to be default data that's contained within R. So hopefully we shouldn't have too many problems with that since they're already contained. Let's see if we can do this one by one. There. So everyone should now have access to the data that we're going to need. And I'm going to hop in to a very brief presentation to orient you to what you're going to be doing. All right. Is everyone able to see my screen? Wonderful. So again, we're going to talk today about publication quality plotting in R, building on the principles that Dr. CarRonna introduced. I want to begin by acknowledging that the location currently occupied by Western Colorado University is the unceded land of the Uncompahgre Ute people. Today, we are going to start by talking about choosing color schemes and why it's so important to choose a color scheme ahead of time. We're going to talk a lot about the principles of ggplot2, first focusing on ggplot on its own as a graphing tool. And then we're going to look at cowplot and gg3d as handy add-ins. We're going to talk a little bit about Plotrix for heat maps. And then we're going to go into the principles of Plotly, which is another graphing package that I like a lot. It allows you to create HTML-based interactive plots. And I use these a lot in my own work. I love them. So let's think about choosing color schemes. So it's really important to keep in mind when you're thinking about colors is that approximately 4.5% of the population has red-green color blindness or color deficiency, which is carried on the X chromosome. Okay, so 8% of individuals who have an XY chromosome overall will have some form of red-green color blindness. Okay, and that's actually higher in individuals with XY chromosomes of Northern European descent. Okay? 10 to 11%. And then about 0.5% of individuals with XX chromosomes will carry this red-green color blindness, right? So that winds up being a pretty fair proportion of your audience at any given time, okay? Blue-yellow color blindness and complete color blindness are other forms, and they are carried on other chromosomes. They're not sex-linked chromosomes, and so they're equally distributed in the population. Injury or disease, or even some medications, can affect an individual's ability to see color. And this can range from mild color deficiency to a severe color blindness. So it's really important to keep this in mind when you're choosing your color schemes. We need to make them accessible. And luckily, that's pretty easy to do. There's been a lot of research on what individuals with different forms of color blindness can see and what colors work well for them. So here's a really great example from Wong 2011 Nature Methods, showing here's the color here on the left to people with common color vision, so normal color vision, and then two of the most common forms of red-green color deficiency, and this is what they see. Okay, so it's really, really important to keep this in mind. Going back to those defaults, I'm going to actually switch my screen. You see that a little bit better? Zoom in just a hair. There we go. Back to those defaults. I really appreciate how much Dr. CarDonna hit on how bad sticking with the defaults are, because I'm going to hit that really hard too. So these are default ggplot2 colors. Not only are they ugly, they are not colorblind safe. So this is what those default colors look like to someone with the most common form of color blindness. Protanopia is red-green color blindness with the red deficiency. Okay, you can see that those top two colors, where here, they're like this ugly sort of camel brown and this green, they're almost identical. And so is this one, just about identical. And these all turn into blues that are a little bit hard to distinguish, right? Those default colors are awful, okay? So this is what it looks like to someone with deuteranopia, which is red-green color blindness with a medium wavelength deficiency. They're even worse, right? They just completely blend together. You're absolutely going to lose that chunk of your audience. And to someone with tritanopia, which is yellow-blue color deficiency, they all turn into these sort of pinks and teals, okay, with a little bit of gray. And there again, you've lost a chunk of your audience. That's really important to keep in mind. So here's what they look like all together, right? With normal color vision, you can tell these default colors apart, but any kind of color deficiency, these three most common kinds of color deficiency, they're just awful. We have to keep this in mind. So there are several R packages, and I'll introduce you to some of them that are specifically designed to be colorblind safe. They only produce color schemes that are safe for the three most common forms of color blindness. But there are also some really great web tools and apps that you can use to check your own figures. And I always do this, always. Okay, so the one that I happen to like a lot and know very well is Chromatic Vision Simulator. So this is available as an app for both Apple and Android, but there's also a web edition where you can just upload a figure and it shows you what it looks like. So this is what that looks like. I have created a figure here with the Viridis package in R that is specifically designed to be colorblind safe. Every scheme they offer is colorblind safe, and I have used the web version of Chromatic Vision Simulator and uploaded it. So I can see side by side what my figure looks like in common color vision, and then to people with protanopia, deuteranopia, and tritanopia. And I can see that with all of these forms of color deficiency, my figure is interpretable still. All right, and so this is an accessible figure. That's really important. Okay, so I'm going to show you how to create these color schemes in R to make sure that your figures are safe for people with color deficiencies. So we're going to do a quick critique of a figure to build on what Dr. CarDonna was saying. Shout out to Rob Ulrich in my lab at UCLA, one of our PhD candidates, for turning me on to this paper. It's otherwise a very good paper. We're going to critique a figure. So this is about the patterns of elemental concentration in shells of invertebrates representing different calcium carbonate polymorphs from the brackish Gulf of Gdansk. Okay, this is a solid paper, but here's a figure that I want everyone to take a look at. Take a minute and orient yourself to this awful figure, and then we're going to talk about why this is a bad idea. Okay, let's critique it. What is one of the first issues that pops up when you look at this figure? Feel free to unmute and shout out. This is the interactive portion. I'm seeing a very pained look on Hunter's face, and I agree.
For me, it's hard to read the difference inside classes. If I really squint, I can see it. Is it 1, 2, 1, 3, 1, 4? It's very hard for me to read that x-axis.
Yeah, I totally agree. It's awful to read. What's something else that jumps out?
Colors everywhere.
Colors everywhere. And they've used the default ggplot2 colors, which we just talked about being terrible. Yeah, what else jumps out?
Some of the letters are overlapping on the points.
Yeah, they're totally overlapping. I can't tell whether these two, like which label each of these points goes with. Totally overlapping. They're awful. What else? Something that immediately jumps out to me is that they have plotted p-values. That's a no. Should never ever treat p-values as if they are a y-axis. Okay, that just don't ever do that. That's not how p-values work, and that is a big thing that jumped out to me. Okay, so otherwise excellent paper, but this figure is a waking nightmare. So with this in mind, we are going to think about how to create better figures that are information rich and valid and actually help orient our audience to our data and our conclusions. Okay. So I'm going to start with a really basic intro to some of the most basic options in ggplot2. Okay. I'm going to go through that fairly quickly, but I know that I've got an audience with a whole different range of experiences with ggplot2. And so I want to make sure that I'm not losing people on the really basic stuff before we start to get into more complex things. So I'm hoping to familiarize you with the grammar of graphics, the GG in ggplot, build awareness of what options are out there, right? I can't show you every possible option in ggplot in just an hour and a half, but I hope to make you aware enough of the code structure that you can then begin to delve in and make your own. And I also want to do this to provide you a whole bunch of code to borrow from for later, because there is nothing more useful than having a bunch of code chunks sitting around that you can just repurpose. I repurpose all of my code. Okay. We're going to then put all of these options together into usable plots, things that you would actually use in a publication or in a presentation, and talk about the best ways to export those in really nice, crisp, high resolution so that they look good. Okay. So I'm going to go ahead and stop my screen share for the moment. As everyone, yes, sorry, Rob, I just saw the chat. P-values on the y-axis, awful. Overlap, awful. Thank you. Has everyone been able to either open up the R script that I sent or get into RStudio Cloud? Okay. Can you put it in chat or give me a reaction if you are ready to hit it? Fabulous. I'm seeing a bunch of thumbs up. Okay. Great. Any issues popping up so far? Okay. If any issues come up, let me know, because I'm facing one of my biggest fears today, and that is live coding in front of an audience, which is awful. So please let me know right away if you get behind on anything, because I don't want to leave anyone. So I'm going to share my RStudio screen with you. Please work along with me, either in local R or RStudio Cloud. I'm going to start a new screen share. Keep in mind that while I'm sharing my screen, it's really difficult for me to see chat. So if you run into any issues or questions, please unmute yourself and just shout at me. Okay. So you should be able to see my screen now. If you are working in RStudio Cloud, I have pre-installed all the packages that you'll need to save time. So you won't need to run this install packages. You'll just need to run library to load them as we go. If you are working in a version of R before 4.0 and you're doing it locally, this could cause some issues, and I highly recommend that you are working in RStudio Cloud. So we're going to start by just loading ggplot2. So R contains a whole bunch of really great built-in data sets. And we are going to utilize a whole bunch of those for graphing. They're really great to practice graphing, and make sure that you're making good figures. All right, so we're going to start by loading the Old Faithful data set. Okay, so Old Faithful, you'll see if I load it up into my global environment, it says promise. That means it's just sort of sitting in memory waiting for me to use it. Okay, if I actually call it by running a ggplot command, it changes from a promise to an actual data set that is now up in my environment that I can look at. Okay, so we have two variables here, eruptions and waiting. Okay, so eruptions be the length of time of an eruption, and waiting is the waiting time in between eruptions. And so we want to look at the relationship between how long the eruption is and then the time you have to wait until the next eruption. So what I've just created is a base ggplot with no customization. And Dr. CarRonna was kind enough to hit on why it's a bad idea to use base graphics. So we're going to delve into that a lot deeper. Okay, so this is just a really basic scatter plot. So the structure in ggplot is that I have a data call inside of this ggplot, and then I have an aesthetics call. This AES is my aesthetics call, and this is where I define my mapping. So I'm telling ggplot I want eruptions on the X, and I want waiting time on the Y. Very simple. And then I use a plus to string it together with the geometry that I want. And in this case, I want a geom point, right? But if I start to type geom, all of these options come up, right? We can do any of these kinds of plots, density plots. We can add error bars. We can do a dot plot. We can do a histogram, a line range, a point range, polygons, everything. I use ggplot for mapping a whole bunch because I love it. We are going to start with some fairly basic ones, right? So this is geom point. It's just a really basic scatter plot. Okay. If I run my next line of code, I customize my labels, and there are a few ways to customize labels. This is just one. Okay. So this labs labels, so X equals and Y equals, really simple, and they're just separated by that comma there. And so that customizes my labels, right? It's my first step. We're going to add color. So there are a number of ways to add color in ggplot. If I put my color equals inside of geom point, I get just one solid color, right? We get this color cornflower blue. And I've put a link here on line 26 to a cheat sheet for the color names, right? So you can call colors by names, by hex code, or by using an external package to do it. We're going to start with color name just because it's the simplest, okay? So now I have solid blue dots and I have customized labels, okay? So everything you do in ggplot is just building one call after another. Now, if I want to color the points by value, instead of putting that inside of geom point, I have then moved my color call into this aesthetics, right? So we're inside of the aesthetics call, and I'm saying color equals weighting. So instead of giving it a color name, I've told it I want to color by the variable weighting. Okay, so this is how long I have to wait. If I run that, okay, it gives me a continuous color scale, and it colors the points by the time you have to wait, okay? Now we could easily reverse this if we wanted to. We're not gonna stick with this color scheme, just to show you how it's done. And you can use this for any kind of geom, right? Color by weighting. Well, now we can customize the legend. Our legend is called weighting because that is the exact name of that in our data set, but that's not very nice, okay? If I go back to this labs call, I can tell it my color is actually minutes. If I run that, it just changes the name to minutes. And that's much nicer. We can make that anything we want, but that's much nicer. Now the legend on the right is a little bit weird for a plot like this, right? So I can keep everything the same and add a theme, right? So there's a whole bunch of theme options in R. Well, if I want to move my legend to the bottom, it's theme legend.position equals bottom. And you can put this anywhere, top, left. You can turn it completely off by using none. But if we just want to stick it on the bottom of our plot for now, that's what you do. Okay. Well, as Dr. CarDonna was talking about earlier, this default background, lack of lines, that's all pretty awful, right? It's ugly, it's not informative, it's distracting. So ggplot actually has a whole bunch of built-in themes that are meant to be journal compatible. Theme classic is actually what most journals will ask for. Well, that removes that awful gray gridded background and gives us these nice crisp lines, right, on the X and Y axis. And a lot of journals want to see that. Note here that I have switched the order. I have put theme classic before theme legend because this theme classic will overwrite that legend position call, right? If I switch the order of those, theme classic will move the legend back because that's where the default is. So I've changed the order. Another thing that you can do is change the base font size. Inside of any of the calls, the geom calls, you can change the size of points. You can change the size of labels, but if I want to change the base size, so like every font size in the whole plot, I put that as base underscore size inside of my theme. Okay, so if I were to run that, all of a sudden my text is huge. Okay, so that's one option. We can also change the base font, right? Base family is serif, right? So the default base family is just a sans serif font. But if I want it to be more like Times New Roman, right? Base family equals serif changes that font, and we can see how different that looks, right? Most journals want a sans serif font, but you might want a serif font for some other purpose, and that's fine. Okay, so that's how you do that. You can also change these line sizes, right? If you want it to be heavier, if you want it to be lighter, you can change it using this baseline size, okay? I'm gonna change it. I'm not saying that's a good idea, but I want you to know that that option is available, okay? So those are just a few of the basic options. So within any of these things, there's a whole bunch of stuff that you can change. So if I start to type theme, a tooltip is going to pop up with all of my theme options. Right? You can change literally everything about a ggplot, and this is where you would find it. Okay, you can choose any of these. Backgrounds, plot tags, you can add a panel grid, you can remove panel grid, you can change absolutely everything, spacing. So the purpose of this really quick basic introduction is just to familiarize you with the structures so that you can then customize your own plots as you need to. But to make a really nice publication quality plot, we're going to ditch all of those really weird options I just showed you and go back to something a lot more simple. Okay. So change my color to blue 4. Okay. Just a standard dark blue, right? You can change this to anything you want. If I change it to one color, remember that the legend is going to go away by default, and then I no longer need this legend position argument. Okay, so I've removed the color equals weighting out of my aesthetics call, right? It's just X and Y now, and I've made it one solid color. That gives us just this very simple scatter plot with blue dots. If we want to add a trend line, there's lots of ways you can do this. One of the really easy common ones is this geom smooth, and it adds a smooth. If I do method equals LM, right, that's just a straight line, it's a linear model. There are lots of different options too. Okay, using the formula y by x, and it gives us a standard error around the line by default. Okay, if I don't want that standard error, and in many cases I don't, I can go SE equals false, right? And if I run that, it takes that gray band away and just gives me a line. It's pretty ugly, right? That blue color doesn't work with the other blue color. And I specifically picked a point that default blue wouldn't work with, so I can show you your options, okay? So if I go into Geom Smooth, I'm going to stick with method LM for now. I can just change the color of the line to black, and I'm going to add an annotation as well. Okay, if I run this code, if I add annotate, Geom text is telling it I want to put just text somewhere on the plot. The X and Y position of your annotation need to be set. And I'm showing you here two different ways to do it, right? So we're using the X-axis range as a way to set where we want our annotation to go. So I'm telling it, find the minimum length of an eruption in our whole data set and then add 0.5 to it. Okay, so you can try changing this. If I add 0.1, moves it to the right. We're taking the minimum plus 1. We can say minimum minus a half, and it moves it a little bit off screen, but it also tries to adjust this x-axis for you, right? So we're going to go back to minimum plus a half, and the y I've set at 90. I want it right in line with this, but you can set that as math too, right? Maximum point minus a half or something like that. And I've asked it to give me red text just so that it pops out to you so that you can see it really easily. Okay. But we want real info here. So we're going to create a model for the length of eruption and the wait time between eruptions. And we're going to extract some of the model fit info to use on our graph. So for a basic linear model, which is all I'm doing here, I've done a couple of things, right? So I've done the LM, so y by x, right? So the length of the eruption is predicting the time we have to wait to the next eruption. Data equals faithful, our built-in data set. I've wrapped all of that in a summary command, right? So I can just select that little bit of data, or that little bit of code rather, and ask for the model summary, right? If I just highlight what I want, it'll run it for me. Okay. So we can see that we've got some model fit statistics here. Our multiple R squared is right down here, adjusted R squared. Well, we want to extract just that R squared. So I have taken the summary and I have wrapped it in the round command, right? We don't need the like 30 digits or whatever it's going to give us. And so I've told it, give me the summary. I've used this $ adjusted R squared so that we just extract the adjusted R squared from our entire model fit summary, and we're going to round it to 2 digits. Okay, so that's what this line of code is doing. And if I run that, it tells me my adjusted R squared is 0.81. Okay. Now there are more sophisticated ways to do that, but to keep from delving into like non-graphing kinds of codes, I'm trying to make this as simple as possible. So that if I want to add this, instead of saying model fit info, I want to add this adjusted R squared of 0.81, but I want to format my code so that I actually get the R superscript 2, so for R squared, right? So if we want to add those kinds of things to a label, we come down here to our label and we make it an expression. And to do what I want to do, I'm going to use expression and then paste and then my label. And I've put the R caret squared and then an asterisk outside of my quotation marks. And then in quotation marks, my equals 0.81. Okay, and so it makes a label that looks like this. And it's going to give me this is in A applied to a list, like that's fine, it doesn't matter. It's just letting you know that that's what's up. Now it says R squared equals 0.81. It's exactly the label I wanted, and that's what's achieved using expression paste. And you could mix and match, right? You could do subscripts, superscripts, multi-part labels, anything you need to do. This expression paste is super, super handy, okay? But there are also some really great external packages that will allow you to extract your model coefficients and put them on the plot without you needing to do any of that work. And this is where the package GGP MISC comes in. It's miscellaneous. It adds a whole bunch of miscellaneous functions. I'm going to go ahead and load that. It has a bunch of cool functionality. The one piece of it I'm going to show you is this adding equations to your plot, but I really encourage you to explore this package because it's really powerful. So we're going to start by telling it what our formula looks like, right? If you had a more complicated formula than just y by x, you could write that in here. But we're telling it my formula is y by x. So instead of annotate, which will now conflict with the GGP MISC that we just loaded, I'm going to use the stat poly equation, okay? If I hold my mouse over it, like anything else, it's going to show me what my form needs to be, which is super duper handy. This starts with an aesthetics call, right? Our label equals, and we're going to paste. Remember, we saw paste above. Equation label, our squared label, and our separator is spaces. So we want 3 pixels in between, or 3 spaces in between. Okay. I'm going to give it a label position, right? I want my label on the X at the left, on the Y at the top. And you can change that if you want it at the bottom right or in the center, right? You can change all of that. Formula equals the formula we just gave it so that it knows what order to look for things in. Parse equals true, meaning it's going to separate those pieces out instead of cramming them all together. It's going to actually parse what's there. Size equals 3, right? Just a text size. If I run that, it's going to put this label. So we've got our just a standard linear formula, right? Y equals mx plus b, except it's flipped that around. It's completely fine. And then our r squared, same r squared that we had before, right? Because it's extracting it right out of our model summary. So that's a super handy package that I really encourage you to spend some time exploring. So now let's think about exporting this figure, right? If we're happy with this figure, maybe we want to change this up again. We're now looking at black points in a blue bar. If we're happy with that, I would not necessarily publish this, but just to show you, put this back to false, right? I don't really need that center error. It's not telling me a whole lot. So there are several ways to export, right? I'm sure everyone is familiar with this little export button. Save as image, copy to clipboard, save as PDF. Well, the default export using this button is 72 dpi, which is really just grainy and awful, right? It's unusable. It's fine if you're exporting a figure just really quickly for yourself to make sure it looks okay on export. But journals are going to want usually 600 to 1200 dpi, and 300 is probably a minimum, an absolute minimum for presentations, okay? We use ggsave for that. So ggsave automatically exports whatever you plotted last, okay? Whatever ggplot you plotted last. You can tell it the format you want by using just the file extension. I use TIFF most of the time because I can do lossless compression, and most journals want TIFF, but you can use practically anything you need. And I'm going to tell it, I want a DPI of 300, right? And then if you run that and give it a second to save and then go into whatever your working directory is for this project, that figure is going to be there, and it's going to be a higher resolution. And it tells you your plot is 4.31 by 4.39. Now, if I want to specify a height and width, which I usually do, right? If I know that the journal that I'm going to submit to wants a figure width of 3.5 inches, which is a standard column width, I can set that. If I want it to be a full page width, I can set that, okay? I want this to export with equal axes. And so I'm going to tell it I want a width of 6, a height of 6, and my units are inches, right? You can use pixels, you can use millimeters, whatever you want. And I'm going to set my compression. TIFF files tend to be huge, and I can actually compress it using LZW, which is a lossless compression format. You're not going to lose any resolution. I'm going to give it 300 DPI, okay? And it'll just save that for me. And if you were to open up your working directory, you could see that that file has been saved. I could ratchet up that DPI as high as I need it to be. And that compression is going to help keep my file size down. And that gives you really beautiful plots to work with. But we want to move on from a basic scatter plot, right? Most of us don't encounter basic scatter plots all that much. But before I move on, I want to pause and see if anyone is having any issues, and make sure everything is running okay. I can't see everyone's pictures, so just give me a shout out if you're having any trouble, want me to pause, want me to go back to anything.
I have a question with that GG save.
Yeah.
When you're doing like a set width and height, for example, like 6x6, if I had something that was like, instead of being a perfectly square plot, I had something that was rectangular or wide or long one way or the other, would it automatically compress that so like whichever was bigger, for example, if it was wider, that fit within the 6 inches, and then keep the, like, is it changing my plot the way it looks to compress it into that size?
Yeah, thanks, Hunter. That's a great question. It will force it to whatever size you want. So what you can do is that if you know, well, I want this to be a full page width, but I want the height to be relative to that, you can just set one or the other. So if I say, give me a width of 9 and just let the height be whatever it's going to be, I can do this. So, and it's made it a 9 by 4.39 plot. It'll scale the either the width or the height to whichever one you actually specify. It's a great question. And we're going to actually use that later when I show you how to make kind of a wide panel plot. I'll show you how to allow it to set the height to match the width. Great question. There's another option you can use with ggSave also. Sometimes you'll make a plot that looks really, really crowded in your tiny little plot viewer over here. But when you export it, you want to scale it. So this is just a straight multiplier, right? So if your text is a little bit crowded, but you need bigger text, you can scale it and it'll make it just a little bit bigger. So this is 1.25 times whatever the default is. And now it's giving me a plot that's a little bit bigger, and it'll shrink my text relative to everything else. Okay, so that's another option that you can use. Okay. So now let's move on from the basic scatter plot. We're going to use plant growth data. Okay, so this, again, you can see that it's a promise. This is an experiment. Our variables are dry plant weight in grams and a treatment group. So if I just do a super simple box plot, you can see that now that's gone from a promise to an actual data set in my global environment. Click that. So we've got a plant weight here in grams and a treatment group. So we have a control and then 2 treatments. Well, again, like box plots can be super information rich and really, really nice, but the defaults are hideous. They're just awful. We never ever want to stick with this default, just like Dr. CarDonna was talking about. Okay, so I'm gonna show you a different way to add axis labels, right? Like anything in R, there's a million ways to do things. And so I'm going to show you a couple of the really common ones, and then you just use whatever you like best. So instead of doing labs all in one call, I'm just going to go X lab, treatment group, plus Y lab, dry weight in grams, right? Just another super simple way to customize my labels. Okay, well now I want to customize my tick marks, my tick axis labels, right? Right now they say exactly what's in the data set: CTRL, TRT1, TRT2, right? It's not good. I don't want to stick with that. So what we're going to do is add this argument here. The scale X discrete, right? We have our scale, which is the X axis. Okay, X discrete, meaning we have discrete variables. You can use scale X continuous if you have a continuous variable like we have on the Y. Okay, so all I'm going to do is say my labels, and C means concatenate or combine, right? So in the same order that my data are written, I'm going to say control, treatment 1, treatment 2. It's all we have to do to customize those labels on the bottom. Really, really simple. It's very simple if you have a few. I'm going to show you a way later to customize everything in a really big set of data. If you have a whole bunch of discrete labels, I'll show you that in a little bit. But for now, we're going to stick with these fairly simple ones. So now we want to understand if there are differences between the groups, right? This is an experiment. We have a control and 2 treatments. Okay, are there differences between them? So I am going to do a standard ANOVA, right? AOV. And again, we've got Y by X. Is there a difference in weight by group? Data equals plant growth. Okay, and I'm going to take my ANOVA and just wrap it in my post hoc test, the CarDonna significant difference, which those of you who have taken stats will probably be familiar with already. If not, super handy to look up. It's a really nice test to use, right? So we're going to look for pairwise differences between groups in a really basic ANOVA. If I run that, you can see at the bottom here, I get this adjusted p-value, a lower and upper, a difference, and then each of these is pairwise, right? Treatment 1 to control, treatment 2 to control, treatment 2 to treatment 1. Well, after adjusting for multiple comparisons, the only significant difference I have is between treatment 1 and treatment 2. Okay, neither of them is different from control, but I do have a difference between treatment 1 and treatment 2. Well, I want to show that on my box plot to make it really clear to my audience what's going on. So to do that, I'm going to add annotations. But I want to note that because we have loaded that GGP MISC package, it's going to conflict with annotate. Okay, so what I want you to do is come down here to annotate and add this. ggplot2 : annotate. And that is going to force R to use annotate from ggplot2 instead of allowing it to conflict with GGP MISC, right? And now if I run that, I've got labels, right? So my control is both A and B, right? Treatment 1 is A, treatment 2 is B. And that's showing treatment 1 and treatment 2 are different from each other, but neither is different from control. And now we've added these labels, right? So Y 6.5 means I put it at right at the set 6.5 marker at that line. Okay. And then I'm using the same X that we have in the data. Note that I am not using the new X label that I've set because that label does not affect anything in the underlying data. Okay. It's just a label. So I'm sticking with what's in my data set as the group, right? Our factor, control, treatment 1, treatment 2. Okay. And that's given us A, B, A, B. All right, well, let's make it look prettier. I'm going to add that ggplot2. Okay. And now we're going to add a theme to it, right? We're going to add this theme classic. Because again, we don't want base stuff. We're also going to fill by group. I always try to use color whenever I can, even if it's really obvious that they're different. It does really help the reader. It helps visual interest to have color in your plots. Now again, these are base ggplot2 colors, and they are awful. They're terrible. So I'm going to show you some really nice ones instead. And again, remember, we're going to go in here, add that ggplot2 so that we're not conflicting. We're going to move to the Viridis package that I told you about earlier. Well, we don't want those awful, awful default colors. We're going to get nicer colors out of Viridis, right? And again, if you haven't got Viridis installed, go ahead and install that now using this install packages. But if you're working in RStudio Cloud, you can just load it because I installed it for you already. So note, when we are doing box plots, we are setting a fill instead of a color, okay? So if we change this to a color, let's see what happens. Okay, and that can be nice in some circumstances, right? You might want to do that. But for our purposes for right now, we're going to go fill. Okay, we're going to leave the default sort of black color here because it's a nice contrast. And we're going to fill by group. But now what we're adding is the scale fill Viridis D, and D is discrete. Okay, so there are discrete and continuous options for both color and fill, which is super nice. We're going to leave it all at the defaults by leaving these parentheses empty. Okay, so that gives us a fill. Well, this is colorblind safe, but it's pretty ugly. The default is pretty ugly for this particular application. First thing we're going to do, though, is remove that legend, right? It's really obvious what maps to what. We don't need this legend. It's just going to clutter things up. So I'm going to add at the end, it goes after theme classic, otherwise theme classic is going to overwrite it. Theme, legend, position, none. Okay, legend's gone. So as Dr. CarDonna was talking about, making figures really is an iterative process. And so I go through this with my own figures. I'm going to add one thing or two things at a time where I know what their effect is going to be. Okay. Now I'm going to change my Viridis options. Okay, so if you start typing scale, fill, a bunch of stuff is going to come up. Continuous, date, time, right? But if I type Viridis, all of these things are going to come up. I'm going to hold my mouse over Viridis D, which is the discrete option, and it's going to show me all these different things I can do. I'm going to select that and type option. Okay, and a tooltip pops up. So we have magma, inferno, plasma, Viridis, which is the default option, and civitas, right? So all of these palettes are colorblind friendly, but they have different optical properties, right? They're going to look a little bit different. So we're going to start with E, and I'm also doing this alpha. Alpha can be added to any geom, and this is transparency. So an alpha of 1 is completely opaque, and an alpha of 0 would be totally transparent. So I'm going to set it at 0.7 or 70% opacity. And I'm with option E. It's pretty nice looking, right? I think that that's the visually appealing color scheme. Let's try changing the alpha. If we make it 0.4, right, makes it even more faded, right? And that doesn't matter because nothing is behind it. That's going to start to matter if you make figures that have points overlapping or curves overlapping, which I will show you, okay? So what I want you all to do is take a moment and play with these different options, right? What does C look like? Okay. And again, all of these are designed to be colorblind friendly, which is great. Okay, we're going to change this to a 7, make it darker. Okay. Just meant to choose Inferno. Okay, option equals, oh, Inferno is B. Okay, that's what Inferno looks like. Okay. Everyone able to get that to work? Anyone stuck getting the Viridis options to work? Cool, I'm seeing a couple of thumbs up. Thank you for that. Awesome. So this is really nice for generating pretty usable, accessible color schemes. And it's also a lot less painful than manually specifying a color scheme, which I have certainly done. Okay. So now, over, I'm going to change that back to E and an alpha of 0.5. Okay. We're going to use the same data, right? The same data and make some density plots. And then we're going to come back to both of those plots later and I'll show you how to put them together. So now we're moving on to making these density plots. I love density plots for a lot of the work that I do because it's a really nice alternative to a histogram where maybe a histogram doesn't look as good or isn't as informative. So we're going to use the same data, plant growth. Note that my aesthetics, instead of being X equals group, Y equals weight, and fill equals group, right? I have dropped the Y argument, right? X is going to be weight, fill is going to be group. We have no Y argument because it defaults to Y being your density. Just like it would be a count in a histogram, this is going to be a density. We go geom density, and I'm filling by group using the same color scheme that I have specified here. Okay, we're using the exact same color scheme. And now that gives me a density plot. And because I have my alpha set at 0.5 at half opacity, I can see behind them. I can see how the curves overlap and interact with each other. And it's given me a legend by default, right? and we know we want to customize that legend, I can do that right inside of the scale fill call, right? So my scale fill Viridis deep, I'm going to name it group, and I'm going to tell it my labels are this. Okay, I can do that right inside the scale fill. And if I do that, it's a lot prettier. Okay, that looks much nicer. But we really don't need this legend on the right. It's kind of squishing our plot and detracting from our main point. So what we're going to do is move the legend to the bottom, and we're going to make it a horizontal legend this time instead of a vertical legend, right? Legend direction is one of those theme options that you've got available to you. We're going to move that legend to change its direction. Then it's going to look like this, right? If I remove this legend position horizontal and just make it a legend at the bottom, sometimes this is going to matter and sometimes it's not. So in some cases, especially with a continuous legend, it's going to try to put labels up on the top instead of at the left, and legend position or the legend direction horizontal will fix that. It's defaulting to horizontal, but I wanted to show you that that option is there, and you can force it to be vertical or whatever you want. Okay, so it's group. That's a lot nicer, right? We have a legend to orient people, but it's not taking up a big chunk of our plotting area like it was before. Okay. We're going to come back to these in a little bit when I show you cow plot. For now, we're going to move to a new data set. Okay. We're going to go to the hair and eye color. Data set examining hair and eye color of statistics students. So one note, we've got this hair eye color as a promise right now. We need this to be a data frame. So it's in R as like a vector. It's not really usable. So we're going to go as dot data frame hair eye color. Okay, and now it's a data frame in our global environment with hair, eye, sex, and frequency. Okay, frequency that these different combinations occur. We're going to start with just a super basic bar plot. Now we're going to make a stacked bar plot. You can make bar plots that are not stacked, right? Just standard bar plots, but stacked bar plots are often really handy. And we do that by saying position equals fill and stat equals identity, okay? Stat equals identity is key. That's what's gonna line it all up. So if we run that, we get this stacked bar plot, okay? Male and female and black, brown, red, or blonde hair, all stacked up. And the frequency equals to 1 because it's position equals fill, okay? So it's basically percentages now. Well, we want to change all of these things. Okay, we want to change a whole bunch of stuff about it. We want to be inclusive with our language, right? So instead of saying sex, we're going to say gender identity. And Y, we're going to make it frequency instead of just this freak. Okay, and I'm going to show you a new scale fill package called Color Brewer 2. It's such a popular package that it is now loaded as part of ggplot by default. But you can also load the package itself. It's Color Brewer, our Color Brewer. Okay, so if I do that, okay, our Color Brewer is the library. But everything I'm going to show you has been grabbed from our Color Brewer package and made a default part of ggplot 2. So you don't have to load anything else. What's really nice is that Cynthia Brewer, who created this, has a whole website showing you different visualizations, right? So it's a lot of the stuff is used for mapping. But for now, we can be content to just grab this palette, okay? So if I run this, scale fill Brewer instead of Viridis, we're asking for the yellow, green, blue palette, and the name is going to be hair color, okay? Something to note is that we can grab these palettes, but not all of them are colorblind safe. And so you're going to have to pay close attention to what is and isn't colorblind safe. And we're going to add some grid lines to help orient people. Okay, we're going to add y-axis lines. Panel grid major Y, element line, color gray 80. So gray 80 is sort of a pale gray. So I've added a bunch of stuff here, right? So now we've got these stacked bars. This does happen to be a colorblind friendly color scheme that I have chosen. But again, check your figures. It's very, very important. So now we have gender identity on the bottom, frequency here, hair color looks nice. We can stick with the default names out of the data set because they are capitalized, the formatting is fine, right? One thing to note is that this pale yellow looks just fine on a computer screen. Like on my screen, this is perfectly clear, it's fine. This does not work on projectors or in print. Okay, so if you're doing a presentation like I'm doing just from a computer screen and sharing it with people, it's fine, but I would certainly not submit this for an article that I knew was going to be out of print. And I would definitely never use it in a presentation. Yellow, especially pale yellows, tend to just wash out and disappear, and you don't want that. Okay, so I would encourage you to play with all these different palette options, right? And just like before, we can say scale Bill Brewer. Okay, and I can hit F1 and it's going to bring up help. All these different palette options, all these different fill options. Okay, it's going to come down, if you come down here to the palettes, all of these different palettes are available by default through ggplot2. Okay, so I'd encourage you to play with these. There are lots of other options available through ggplot as well. Okay. So now that we have created that, let's move on. Sometimes it's better to do a horizontal plot. Nothing could be easier than flipping these axes. Chord flip will flip your axes. And now we have a horizontal plot. Note that this panel grid major has now changed because we want lines running up from the bottom, right? The X. All we had to do was change that from a Y to an X, and that flips it. Okay. I want to move on now to working with slightly more complex data sets. So before I do, anyone have any questions? Anyone stuck on anything not working? I'm seeing a no from Rob Ulrich. Does that mean that you're not stuck or that you're having issues?
That I'm not stuck.
Okay, perfect. Awesome. Okay, let's move on to working with slightly more complex data sets that are maybe a little bit more realistic for all of us doing ecology and environmental science work. Okay, we're going to use the MPG data set. That's, again, we can see that it is now a promise. Okay, and then once we run something with it, it becomes a data set in our environment. So this is fuel efficiency of cars using a whole bunch of different categorical variables, right? So we have city and highway fuel efficiency in miles per gallon by manufacturer, model, displacement, year, number of cylinders, type of transmission, whether it's front wheel, rear wheel, or all-wheel drive, like everything. Okay, class of car. So this is a bit more complex. We're going to start with a box plot again, right? So you've seen a box plot already. And instead of theme classic, I've given it theme black and white. So you can type theme, and if you start typing, it's going to pop up this list, and you can take a look at all of these options. Okay, theme gray, base size, base family, base line size, and you can modify any of those things that you want. Okay, theme light, line draw, minimal. Theme void for mapping is just a blank canvas with nothing, and you add everything you want to it by hand. Okay, we're going to go with theme black and white for this because I think these vertical grid lines in this case are going to be important to keep people oriented to this really complex set of data. Okay, but our axis labels need to be readable, right? They're all overlapping, they look awful. So we're going to actually just flip them around. So going back to this theme argument, I've set theme black and white first, which is really important because the second theme will, it'll overwrite the second theme if we don't have it in this exact order. Okay, so theme, axis text, X, so that means that's the text right on the tick labels themselves. Element text, and we're going to give it a 90-degree angle. Vertical adjustment of 0.5 and horizontal adjustment of 1 are going to put it right up next to our axis. So now we can actually read that. Still doesn't look fabulous, but we'll work on it. Audi, Chevrolet, Dodge, Ford, right? We can read that. So let's take a look at what these do. If I take the vertical adjustment and set it to 1 and don't change anything else, okay, that has actually moved these to the right. So a vertical adjustment of 1, when you're talking about text at a 90-degree angle, means that the top of the text is going to align with the tick mark. Okay, well, we don't want that. 0.5 is what's going to center it. Okay, so we move it back. Now it's all nice and centered. Horizontal adjustment, when you're talking about this with this text with a 90-degree angle of 1, is what's going to put it on the right-hand side of the word. Okay, so if I put this to 0.5, it moves it way down. Sometimes it can be really nice to add that extra space. Okay, it's moved it down and it's actually centered them with each other. So that 0.5 centers your labels with each other. And that's all based on the longest one, which is Volkswagen. Okay, that can be super nice in some cases. But we want that, for readability's sake, to stay at 1 so that the right-hand side is aligned. And those could be negative numbers too, right? If I want way far off, I'm going to set that to -1. Now, there aren't many cases where that's going to be handy, but just so you know that that's an option, you can use negative numbers. We're going to put it back up here. But it's all lowercase. So I showed you before how you can manually change these labels to be capitalized, right? You just write the label in that you want, right? But with this many, it's going to be a giant hassle. And we don't want to modify the underlying data set, right? So one option would be to go into our data set, capitalize each one of these things, and then reload the plot, reload the data, then reload the plot. What a hassle. We don't want to do that. So instead, R has this function, just base R, called toUpper, and it will take a character string and capitalize every letter. Well, we don't want every single letter capitalized. We want this to be noted, like marked as proper nouns, right? The L and the R in Land Rover should be capitalized, but nothing else, right? So toUpper provides this canned function, not a function I wrote, it's provided right in their documentation. I'm going to just load that function into memory. They call it cap words. Okay, and the way this is written is that it capitalizes the first character, the first letter of each word, and leaves everything else lowercase. Okay, so what we can do is recode the levels of manufacturer, right, our x-axis, to be capitalized. So right now, you can see they don't actually have levels until we give them levels. Right now, it's just as a character string, and so we're going to wrap manufacturer in as.factor. Okay, so we're going to tell it, I want manufacturer to be a factor, not just a string. And I'm going to tell it for every level, right? So for each manufacturer, capitalize. If I run that all by itself, right, without assigning it to anything, it actually did what we wanted it to do. Audi is now capitalized where it wasn't before. Chevy is capitalized where it wasn't before. And Land Rover, right, 2 words, the L and the R are capitalized and nothing else is. Okay, so we're going to go ahead and make that assignment. Note that it doesn't change the underlying data at all. You are just giving it this extra attribute. You're telling it that these are levels and they should be capitalized when I plot them, but you don't change the underlying data. It's really important to avoid changing the underlying data, especially when you're working with complex stuff. Okay, so if I run that, I'm telling it my labels are the levels of manufacturer. Instead of being just MPG manufacturer, here I've defined levels, right? It's a really different thing. So my labels are the levels of the manufacturer. And now it's showing up capitalized just like I want it. So we can actually skip the recoding altogether if you want to, right? Rather than going levels, MPG, cap words, levels, right? All of that. We can just use this cap words part okay? Cap words, levels, as dot factor, MPG manufacturer, okay? It'll produce exactly the same plot. Doesn't matter. You can do it whichever way you prefer. Okay, well now we have decent x-axis labels. Let's make this a bit more information rich. So now we're going to fill it as we're going to do a fill by the number of cylinders. So note that I'm making the number of cylinders a factor because right now it's an integer. It's going to think that that's a continuous variable and give me a color, like a continuous color scale instead of discrete colors, right? But we know that the number of cylinders in a car is not continuous, right? It's 4, it's 6, it's 8. So if I tell it as dot factor, it knows now that we want a discrete color scale, okay? Now again, we could argue that these are pretty ugly colors, right? We can customize all that, not a problem. But we want to make it a bit more usable. We really kind of want, we want a lot of things changed, right? This is information rich, but it's really crowded and it's really hard to read. So it's going to be a lot easier to read if we break this into a bunch of subplots. And that's where facet grid comes in, okay? So facet grid will take whatever variable you give it and break it into subplots. And we're telling it we want to do it by cylinder. Note that I have a little tilde here, which you can read as by. So facet grid by cylinder. And I'm wrapping this again in as dot factor so that it remembers that it's actually a factor. Okay. So now we have this broken into 4 subplots, right? It's much easier to see the patterns. However, we have one manufacturer, so of course Volkswagen has to be different and make a 5-cylinder car, right? That's not really telling our audience all that much. And it's really crowding our plot in a way that's unpleasant, okay? So we can just, in this case, I'm gonna ditch this. I'm not deleting it out of the underlying data. I'm just not plotting it because it's not informative. In my caption, maybe I would say, one manufacturer has produced a 5-cylinder car and this is its mileage, right? It's 20 something. But it's really not adding anything to plot it. So I can actually exclude data from right inside my ggplot call. Okay, so I'm asking it, so give me miles per gallon, our data set name, where, right? And the brackets can be read as where, miles per gallon, $ cylinder, right? So where the number of cylinders is not 5. Okay, so exclamation point equals means is not, right? If I used double equal signs, it would be is exactly, okay? But I'm asking it, give me everything except for where the number of cylinders is 5. And this comma tells it, keep everything else, right? Keep all of my columns. I'm just deleting one row. So if
Anna?
I do that, yeah.
Does doing that also work with non-numeric variables?
Yes, so if I use the is not with a non-numeric, I can say where is not Audi, like
Cool.
that.
Thank-
And instead of a number, I would put it inside of quotation marks. Great question.
Cool, thank you.
Yeah, no problem. Okay, so we're going to tell it it's not 5 cylinders. And now we have a plot that's getting closer to being usable. Okay. Of course, we are going to want to modify colors and names and spacing, right? But you are much closer to a usable plot now. We are beginning to run short on time, and I want to be sure to get to things like cowplot and maybe even plotly. I have given a list of resources here at the bottom of this section. So how to modify components of a theme. So I've been showing you theme, legend, position, and all that kind of stuff. Well, here's every single possible option for theme. Can come in really handy. Here's a really fabulous ggplot tutorial. So a really complete tutorial if you want to work through this on your own. And here is a running list of color scheme packages. So I've showed you Viridis, and I showed you a little bit from Color Brewer, but there are a ton out there. There's a whole Wes Anderson palette. set of palettes that I really enjoy. There are a whole bunch of palettes that are designed around the color schemes of scientific journals. There are a whole bunch of like outdoor themed palettes. There's video game palettes, there's Star Wars palettes, anything you can think of. And so this document has a great running list of everything that's available. Again, not all of them are colorblind friendly, so you're going to have to check them. But these are really great options available to you. I'm going to move on quickly to CowPlot. We're probably not going to get through all of the code that I have written, but at least you have examples of stuff that you can do, of options that you do have, okay? But we're going to start with CowPlot because I want to be absolutely sure to show you how to do this. Okay, so CowPlot is a really nice companion package to ggplot. It can be used with base plots too, with base graphs too, but it's especially powerful when combined with ggplot. So for this, I'm just going to generate 3 sets of random data, right? So data 1, 2, and 3, okay, entirely random. They have an N of 500. So this one has a mean of 1, standard deviation of 1.2, mean of 2, mean of 3, right? We're just generating random data. And these are normally distributed using the rnorm function. We're going to turn those into a data frame. I'm going to say column X is data 1, column Y is data 2, column Z is data 3. That's our example data. And now that they're a data frame, I can plot them. So note, you can always assign a ggplot to an object like you do anything else. I haven't been doing that because I would rather that it just display for you right away. But now we're going to need to assign this to an object. I'm going to start with a very, very simple histogram with 50 bins. Okay. All I'm giving it is an X aesthetic. So X equals my X column. Data equals my example data. Okay, just theme classic, because why not, right? I'm not going to do much more customization with this because the purpose of this is to show you how to arrange plots into a really nice panel. So we just have a histogram, normally distributed data. I'm going to do a second plot with my Y column. Okay, just another histogram. Third plot with my Z column, another histogram. And then let's make a quick scatter plot, a geom point with X and Y. Just a scatter plot. Well, this is where CowPlot comes in. It'll arrange them into a really nice grid for you so that you don't have to go into like Photoshop and try to line them up and fight with it, right? It lines them up for you into this really nice panel plot. Everything is aligned. You can do common X and Y axes if you want to. We can give each plot a label. Like if you're just submitting a panel plot, most of the time you want to go A, B, C, D, right? It knows that. So this labels equals auto gives you a capital letter to the upper left of each plot. That can all be modified. We can do custom labels. We can customize the position if we want to. If we want to do custom labels, again, we use this combine. Labels equals C, and then whatever labels you want to use. I just highlighted something. There we go. And it just puts the labels above for you. Very extremely handy. It also offers these fun stamps. I don't know how usable they are, but they're kind of fun. The good, the bad, and the ugly. On the right-hand side, you can see that it labels good with a little green stripe on the right, bad in kind of a reddish pink, and the ugly in an orange, okay? Kind of fun. But the most practical use of CowPlot is arranging plots with a common legend. Okay, so that's a really annoying thing about ggplot. You might have 4 or 5 plots showing something very, very different about the same data set and need to use a common legend. Well, we can do that. We're going to go back to those plots we made earlier. All right, remember these? Our density plot, okay, and our box plot. Okay, so now I've assigned them to plots 5 and 6, right? Because we need those saved for a CowPlot to get a hold of them. CowPlot can extract just the legend, nothing else, just the legend, and that's called the legend grub, right? So we're going to go get legend out of plot 5, because plot 5 is the one that has a legend. We're going to call it my legend, right? So we just grabbed my legend out of plot 5. So now this is where CowPlot becomes kind of awesome. We're going to turn off the legend on plot 5 when we plot. We don't have to go back into our plot itself and modify it in any way, we can just go plot 5 plus theme legend position equals none. Okay, so if we were to just highlight that bit and plot it, just plots our the density plot without the legend. Okay, so comma P6, comma my legend, and it's going to arrange them into a grid for us, right? Now that could look better, right? The legend is down here at the bottom, it's taking up a bunch of space, it's kind of squishing these two plots. Could be better. Well, we can arrange that using some more options in plot grid. So we're going to use the same thing, right? Plot 5 plus no legend, comma plot 6, comma my legend. But now we're telling it I want it all on one row. And this relative widths tells cowplot how you want it scaled, right? So I want my density plot and my box plot to be the same width as each other, right? So that they are given equal visual weight, but I want to make the legend narrower. The legend I want to be half the width of the other two plots. Okay, so if you run that, see how that looks. Now it's a bit squished in your window, so if you hit zoom, you can sort of resize that window and it's a little bit better. Okay. Now, going back to the question Hunter had earlier, what if you have a plot that you want to be a lot wider than tall? This is certainly a case where that is going to happen, right? I want this to be wide, but I don't want it to be super tall. If this were a square, it would look silly. So I am going to go to my GG save. I'm going to set it to 600 dpi again, because if I'm going to submit this for publication, it needs to be high res. And I'm going to set a width of 9, but I'm going to leave the height free. I'm not going to set a height. I'm just going to let it scale the height to whatever it needs to be. Okay. And again, in inches, and I'm using the LZW compression. And if I save that, it's 9 inches wide and about 4.4 inches tall. Okay. And so that gives me a nice wide plot without it getting squished. Okay. We are getting low on time. So I'm going to leave you to explore the rest of this code. You should all have comments that will help you step through this code. So I have stopped here at the end of line 442 in mine. The rest of this should walk you through how do you make really quick, simple 3D plots in GG3D, which is a really nice extension of the ggplot2 package. If we want to move into non-ggplot2 graphing, we can make heat maps in Plotrix. This is something that I find really handy. Plotrix has a lot of cool functions that I've just begun to explore. I especially like it for heat maps, but it has a lot of other functionality. Okay, so I've provided you an example data set from a paper that we have in review right now. A shortened data set, a smaller data set, so you can make plots. And this steps you through the process of making row names, dropping the column that you don't need, making a palette outside of a ggplot call, right? So you can just make a Viridis palette, how to call that Viridis palette and assign a specific colors to specific groups of data using if-else statements, and then making and saving a basic plot and how to use the built-in plotting device. Okay. Then I also have provided a really basic intro to using Plotly and making interactive plots that you can export as standalone HTML widgets, which is super fun. Okay. Plotly is another really powerful package, and I could probably do 3 or 4 workshops just on Plotly. I love this a lot for making interactive 3D graphics that people can zoom in and out of, select plots or select pieces of it, that kind of stuff. It's really great. It's especially good when you're doing things like shiny dashboards, which I do quite a bit. And you can also, a lot of journals now will accept HTML supplementary information. So for one of my papers, I exported a 3D interactive graph as an HTML widget, and it is supplementary information to my paper, and people can go in and play with the data and really get a feel for what it looks like in 3 dimensions. Okay, so all of that should be there for you to step through, and I'm very happy to take any questions in the last few minutes we have left.
I had a quick question on the differences between facet plot and facet grid, and when to use which one.
Yeah, so they just wrap in a slightly different way. For the most part, they're going to be interchangeable. Facet grid is a bit more flexible in that you can define, like really specifically, make a grid and I want it to be exactly 2 rows and 3 columns. And so I tend to stick with grid.
Okay, thanks.
Very happy to take any other questions anyone might have. Thank you so much for inviting me. This is really fun, and I hope it was helpful.
It absolutely was, Dr. Carroll. Thank you so much.
Yeah.
For everyone that's still here, I just stuck that survey in the link again for you to fill out if you have the time. It's super brief. And also we're going to be getting the recordings from these workshops as well as all the resources on our website, westernms.com. And so if you guys want to go back there and check that out as well, they'll all be available there as soon as we get everything compiled and stick them up. So once again, thank you so much, Dr. Carroll, and thank you so much to everyone else for being here today.
Thanks so much for inviting me. I'll make it so that the our studio code and data remain available to you. So if you want to go back at a later time, you can access that again if you would like to.
That was so awesome. Thank you so much.
I'm glad that was helpful. I had felt like I was trying to rush through a lot in a short amount of time, but
sorry about the
Jenny.
time overlap. We didn't get back right.
No, that's okay.
I learned a lot.
Awesome.
I was like, oh, my graph looks like that. I need to go fix it. Thank you so much. That was awesome.
Oh yeah, I'm glad that I could help. Thanks for inviting me. This was fun.
All right. Take care.
You too.
Thank you.
Bye.
Bye.